Hello everyone, welcome back to InfoScience. Today we are going to discuss the first phylum under the non-chordates, that is the phylum Porifera. Now let us know about this phylum. The members of this phylum Porifera are known as the sponges. These are primitive multicellular asymmetrical marine animals with cellular level of organization. All sponges are marine except Spongilla, which is found in fresh water. Now let us know about its structure. The body of the sponges is vase shape and it have tiny pores all over the body known as Ostia. These pores are the inlet pores through which the water goes in. The terminal pore is known as the osculum, through which the water goes out. The central cavity is known as spongocoel. This cavity helps in the exchange of gases and form a space for the food to come in. Sponges are diploblastic. The outer layer of the sponges are made up of flat cells and this layer is known as pinacoderm. The cells in this layer are known as pinacocytes. The inner layer is known as choanoderm and the cells which are found in this layer are known as choanocytes. So based on the location of the choanocytes, the canal system are categorized into three types. Ascon type canal system, where the choanocytes line the spongocoel. In Sycon type canal system, the choanocytes line the radial canal. And in Leucon type canal system, the choanocytes line the flagellated chamber. Now, let's know about the anatomy of the sponges. So, these collar cells are known as the choanocytes. The amoeboid cells is known as the amoebocytes. So, these are the irregular cells which help in digestion, defense, and transfer of food. The mesohyle is known as the mesoglia, where we find the endoskeleton. So this endoskeleton is made up of spicules and spongen fiber. The spicules is made of two things, that is the calcium carbonate and silicic acid. The spicules which are made up of calcium carbonate are known as calcareous spicules, and the spicules which are made up of silicic acid are known as siliceous spicle. Spongen fiber is made up of spongen protein. So, depending on this endoskeleton, the phylum Porifera is classified. The spicules are of four types: monaxon, tetragon, triagon, and polyagon. Now, let us know about the nutrition and mode of digestion. Nutrition in Porifera is holozoic, means it takes solid food, and digestion is intracellular. Here, the respiration takes place by diffusion through the body surface, and excretion, it also takes place through the body surface, and the unwanted waste is ammonia. So the sponges are ammonotelic. Now let us know about its reproduction. So sponges have two types of reproduction: asexual reproduction by budding. So in asexual reproduction, it is of two types: exogenous, where the bud is formed outside the body, and endogenous, where the bud is from inside the body. And the next reproduction is the sexual reproduction. So sponges have two types of reproduction: asexual reproduction and sexual reproduction. Sponges are hermaphrodite, which means it has both the male and female sex organ. Here the fertilization is internal and development is indirect, that is through the larval stage. Larvae are of two types: parenchyma, where it is solid, and amphiblastula, which is the hollow. Now let us know about the classification of the phylum Porifera. The phylum includes about 5,000 species of sponges, and these are grouped in three classes depending upon the endoskeleton. So the first class is the Calcarea. So in this class, the spicules are made up of calcium carbonate, example Sycon, Leucosolenia. The second class is the Hexatinellida. Here the spicules are made up of salicylic acid, example Euplectella, Hyalonema. And the last class is the Demospongi. Here the endoskeleton is made up of spongin fiber, example Eusponge and Spongilla.